Assalamu alaikum. My name is Hajra Yunus and today's lecture we will discuss about mutation. In the today's lecture we will discuss about exogenous sources of mutation, and these are ionizing radiation, non-ionizing radiation, deaminating agent, oxidizing agent and intercalating agents. Now next is the exogenous sources of mutation. Okay, so uh, exogenous sources of mutations are uh, the external sources of mutation, means something is from the outside. Okay, so if something is from the outside, or if the chemicals are used from the outside, so the chemicals are called the mutagens. Okay, if something which is added from the outside to change the structure of DNA, so these chemicals are called mutagens. These can be chemicals or these can be physical agent. Okay, now the mutagens are classified into physical agents and chemical agents. Physical agents are the ionizing radiations and non-ionizing radiations. Okay, now we will talk about what is the difference between ionizing radiations and non-ionizing radiation. As you can see here, in case of ionizing radiation, energy level is very high and non-ionizing energy level is very low. Mechanism removes tightly bound electrons creating ions, and non-ionizing radiations induces molecular vibration and electronic excitations. Types of radiation. Ionizing radiations are alpha rays, gamma rays, X-rays and beta particles. And non-ionizing radiations are the UV radiations, microwave and radio waves. Now when we comes to the DNA damage, double stranded break. So ionizing radiation may cause double strand break or single strand break, but in case of non-ionizing radiation it's very rare. Okay, and in case of base modification, it will lead to change in structure of DNA, and it's very, in case of non-ionizing radiation it's less common. Now thymine dimers and pyrimidine dimers. Thymine dimers and pyrimidine dimers are rare in case of ionizing radiations, but in case of non-ionizing radiations and basically due to UV radiation, these are very common. Okay, so these are the main differences between ionizing and non-ionizing radiations. Means ionizing radiation is high energy and non-ionizing radiation has low energy. In case of ionizing radiations, X-ray, beta rays, gamma rays and alpha rays particles are involved, and in case of non-ionizing radiation, UV rays, microwave and radio waves are involved. And the most important thing is that ionizing radiations are responsible for single or double strand break, and the non-ionizing radiations are responsible for thymine dimer and pyrimidine dimers, and thymine dimers are more common. Okay, so now you can see the difference between the ionizing and non-ionizing radiations. In case of ionizing radiation, because of X-rays or gamma rays, you can see there is a single strand break or double strand break. And in case of non-ionizing radiation, as you can see here, thymine is present here. Thymine is present here. So thymine, instead of forming bond with adenine, there is a thymine-thymine bond or thymine-thymine dimer is formed because of UV light. Okay, when the UV light falls on the DNA strand, so instead of formation of thymine adenine or thymine-thymine dimer is formed, and it's a covalent bond, so it's not easy to be broken down. Next is non-ionizing radiation. In case of non-ionizing radiation, as you can see here, it's due to UV radiation mostly. Other radiations are also involved, but mostly it is due to UV radiation. Cyclobutane pyrimidine dimer, it is also called. One name is thymine dimer here, another name is the cyclobutane pyrimidine dimer, and it is also called 6,4 photoproduct. Why it is called 6,4 photoproduct? Because here between carbon number 6 and carbon number, it's the carbon number 6, and here the carbon number 4. Okay, So between carbon number 6 and carbon number 4, covalent bond is formed. That's why it is called 6,4 photo product. As you can see here in this diagram, when the UV light falls on the DNA strand, so between the two thymine dimer is formed. Okay, there are other types of also dimer like thymine dimer, thiamine, cytosine dimer, cytosine dimer, but thymine dimer has more affinity compared to other dimers. Next is the chemical agents. Okay, in the physical agents we discussed about ionizing radiations and non-ionizing radiations. Ionizing radiations are basically due to alpha, beta, gamma and x-rays, and non-ionizing radiations are due to UV light, microwave and radio wave, and basically it is due to UV. Next is base analog. What is base analog? A base analog is a chemical compound that is very similar to the normal bases found in the DNA. Okay. Because of their similarity, base analogs can be mistakenly incorporated into DNA due to replication, because the base analogs look like DNA normal bases, so they can be incorporated into DNA during replication. As you can see here, the example of 5-bromouracil, it's the keto form. Okay, and you know that the adenine forms bond with thiamine. Okay, and now because 5-bromouracil, you can, it looks like thiamine. Okay, but it can pair with adenine or with guanine too. Okay, so this is the means 5-bromouracil has the, it mimics like thiamine or it can mimic like guanine. Sorry, it can pair with adenine or it can pair with guanine. And if when the 5-bromouracil is present in the keto form, it will form bond with adenine, because here at that time it mimics like thiamine. Okay, and here when the 5-bromouracil is present in a anol form, it will form bond with guanine, because at this time it is mimicking like cytosine. Okay, so in chemical reagents, base analogues are the chemical compounds that are very similar to normal bases. Okay, so they can be mistakenly incorporated into DNA strand. Next is the guanine methylation, another type of mutation. And in case of guanine, as you can see here, the guanine is present, and if there is a harmful chemical there is a harmful chemical and the chemical has the methyl group, so as you can see here, it will lead to the methylation of guanine. So you can see here there is a addition of one methyl group. So it is another form of mutation by chemical. Another is the deaminating agent. Okay, in case of deaminating agent, you can see here the cytosine is present and here amino group is present. You know that in the deamination, amino group is removed, and you can see here the cytosine is converted into uracil, and first cytosine forms bond with guanine. Now cytosine is converted into uracil and it will form bond with adenine. As you can see that because of the removal of amino group, the affinity has changed. So this is the deaminating agent. If any deaminating agent is added or is added in the cell, so in that case it will convert the cytosine into uracil, and now uracil will form bond with adenine instead of guanine. Next is oxidizing agent. Oxidizing agent, these are the, as you know that the in during the stress condition and other stresses, reactive oxygen species are formed. Okay, so as you can see that there is a oxidizing agent. So 8-oxoguanine is present. It is present in two form, that is anti form and syn form. Okay, when is the antiform? Antiform is when the sugar phosphate backbone, you can see here the sugar phosphate backbone is oriented away from base. Okay, And here you can see the sugar phosphate backbone is away, so it is the anti, and you can see here the sugar phosphate backbone is not away or base is not away from the sugar phosphate backbone, so it is the syn. Okay, now if 8-oxoguanine, it's the oxidizing agent. Okay, and now if 8-oxoguanine is present, so it will form bond with cytosine, and if the 8-oxoguanine syn form is present, so it will form bond with adenine. So if the oxidizing agents are present, so it will also affect the affinity of nitrogenous bases. As you know that the guanine forms bond with cytosine, forms bond with guanine, and adenine forms bond with thymine. So because of the oxidizing agent, you can see the affinity has changed. Next is the intercalating agent. Intercalating agents are chemical compounds that slide between adjacent DNA base pair. So the examples are proflavin, acridine, and ethidium. Okay, so as you can see that here, so it's the proflavin and acridine, and the intercalating agent uh, will intercalate in the DNA strand and will lead to the deletion or duplication or addition. Okay, as you can see here, it's the normal strand of parental strand, and here there is a process of replication. As you can see that here the acridine and intercalating agent is present here. So due to the intercalating agent, you will see that it will lead to either the deletion in the top strand or insertion in the bottom strand, and in the end you can see the daughter DNA strand, one is short and one is long. Okay, so it will also lead to the or affect the open reading frame of the structure. It may lead to increase the length of the strand, or it may decrease the length of the strand. So if we summarize the today's lecture, in today's lecture we discussed about exogenous sources of mutation, such as physical agents, and physical agents are ionizing and deionizing or non-ionizing radiations, and then chemical agents are base analogues, and which are similar to the normal bases in the DNA strand. And then we discussed about oxidizing agents, intercalating agents, and the deaminating agents. I hope so you understand the lecture. If you have any questions, you can write down in the comment section. Thank you so much.